diagnosis of the disease. I am Dr. Rama Gaur. I have done my PhD in chemistry from IIT Roorkee. My expertise is in the synthesis of nanoscale materials by different methods to tune their properties by varying their shape and size, and then ultimately to use those nanomaterials for applications in sensing and catalysis. This is the flow for today's presentation. To start with, we will be talking about what are nano biosensors, what are the applications of nano biosensors, what is the role of nanotechnology in biosensing, the types of nano biosensors, and towards the end, we'll talk about the career and job perspectives in the field of nano biosensors. The highlights of this presentation will be the relationship between disease diagnosis and treatment, what are nano biosensors, their working principles. We'll look into certain case studies where the nanomaterials have been used as biosensors for the diagnosis of disease. And as I said, towards the end, we will talk about the career and job perspectives. So to start with, it's very important to understand the relationship between disease, diagnosis, and treatment. It is very important for us to understand the causes and symptoms of the disease so that we can go for a timely treatment and the best and the most accurate treatment can be provided to the patient in need. For that, it is important that we should have an on-time, accurate, fast, and early diagnosis. Otherwise, the treatment would not work. According to Cambridge Dictionary, an illness of people, animal, plant, caused by either infection or failure of the health rather than by accident is termed as a disease. The disease can be contagious, can be infectious, it can be common, rare, incurable, and a fatal disease. In general, it has been observed that most of the disease hamper the normal body function. They are either caused due to the imbalances in the hormone in the body.
Ma'am, you are not audible.
I am not audible. Let me check once. Am I audible now?
Yes, ma'am.
Yes, ma'am.
No, ma'am.
Yes, ma'am.
Yes, ma'am. You are audible.
Okay, thank you so much. For those for whom I'm not audible, I would request you to join the meeting again. So I shall continue with that. So like I am saying that the diseases are either caused by the hormonal fluctuations or the malfunction of the organ in the phase of disease may lead to the fluctuations in the hormone. या तो hormone को fluctuation की वजह से disease होगी या disease होने की वजह से there will be certain secretions or there will be some fluctuation in the hormone. In general, what is observed that the first symptom of the disease is either a high temperature or the body pain. But it has also been observed that the appearance of these symptoms is quite late and it makes the treatment very difficult. So if we have technologies for the early diagnosis, it will help us for the better treatment and the easy recovery of the patient. These are some common signs of the hormonal imbalance where the increase or decrease or slight fluctuations in the hormonal level has caused anxiety, arthritis, fatigue, depression, hair loss, amnesia, headaches, and even weight loss, to name a few in the long list. So slight fluctuations are causing a long list of disease. Similarly, when there are disease happening in the body has its own mechanism to give response and to give signal in the terms of biomarkers. And these biomarkers can be again those hormones and enzymes which are already present in the body. Their levels can drop or increase, which gives an indication to us that there is something wrong with the body. So I can define biomarker as a measurable indicator of severity or the presence of some disease. So whenever we go for testing, so we look at the not only the presence and absence, we also look at the quantification of that biomarker to understand that how severe the disease is, what is the level of the seriousness of the disease. So we have a complete list of biomarkers. These biomarkers are helping us to understand what is the state. These biomarkers can be found in different body fluids. So generally, whenever you're not well, you go to the doctor. The doctor always suggests some test. It can be a blood test, it can be a saliva test, urine test, or maybe a stool test. So different body fluids will have these biomarkers. So if we analyze these body fluids with the help of certain techniques, we can go for an early and accurate diagnosis. On the right hand side, you can see a table where there are certain biomarkers which have been detected in the human breath, and the analysis has been done. So these are nothing but simple chemical molecules like ammonia, acetone, and these are very simple molecules: isoprene, methane, hydrogen sulfide, nitric oxide, ethane, and pentane. And in the last column, then you can see there are several serious diseases which have been mentioned: kidney failure, brain seizures, hypercholesteremia, even the airway inflammation, the breast cancer. So all these diseases can be also detected by simply doing the breath analysis. And these small biomarkers will give us an idea that what is wrong with your body. Now, I said disease are happening because of some certain reasons. And when the body is under the state of stress and disease, it behaves abnormally and releases some biomarkers. We go to the doctor, we seek some treatment. But this treatment is not possible if there is no accurate diagnosis. So the main link between the happening of the disease and the treatment to eradicate it is the diagnosis. So we can say that diagnosis of a disease is as important as its treatment is. Any delay in diagnosis can cost the life and the recovery of the patient. Early diagnosis is not only important because it is going to improve the effectiveness, but also it will avoid the long-term implications. So it is very important that those slightest concentrations of the biomarker which go ignored in the initial levels. Whenever you have a fever, for the first 5 days, there was minimal concentration of those biomarkers. You could not detect it in the test. You could not detect whether you had typhoid or malaria or dengue. So if I can develop sensors which detect the slightest amount of the biomarkers, I can go for the early diagnosis. And this is the field where nano biosensors are going to help. So again, it can be said that the identification of the nature of the illness or the other problems by the examination of the symptom is diagnosis. So this diagnosis is a thorough analysis of the facts or problems so that I get an understanding of what is wrong with the body, which part of the body is under stress and which needs attention and treatment. This helps the medical practitioner to plan the treatment approach and provide them the best approach possible such that there's a minimal damage to the body. So if we talk about the types of diagnostic approaches, there are 3 types of diagnostic approaches. The first one is clinical diagnosis. The clinical diagnosis is based on the medical science and the symptoms that has been reported by the patient to the doctor. Like you went to the doctor and you said that I have rashes on my bodies, I have a sore throat, I have stomach ache. So based on those symptoms that you are telling, he will try to correlate the symptoms to what he knows, and then he'll do the clinical diagnosis and he will give you the medicine. In the second case, if the clinical diagnosis doesn't work, there's lab diagnosis, which is simply based on the lab reports or the test that has been conducted for any bodily fluid. Can be a blood test, can be a urine test, can be even an ultrasound. So there are certain tests which are conducted, we get a report, and based on the report, we start the treatment. That is called laboratory diagnosis. The third diagnosis is called radiological diagnosis, in which the medical imaging techniques are used. For example, you have a fracture, you don't have a lab test for that. You will go for an X-ray imaging. If you have some problem with the CNS or nerves or brain, you go for an MRI. So all those techniques which involve imaging, medical imaging, or use of X-rays and mapping are called radiological diagnosis. So among these 3 techniques, lab diagnosis is the one which involves the use of biosensors. or bio nanosensors so that we can detect the problem at the source or at the earliest possible. So now when I'm saying, okay, we'll be using biosensors, there are certain questions that we should ask to ourselves, and we'll try to answer and discuss those questions. The first question is, how do I ensure early diagnosis? There will be certain secretions and slight fluctuations which go unnoticed in the initial phase. And when the symptoms appear, it's too late. So it's again, the first agenda for us is to go for an early diagnosis. The second one is, I also have to develop highly sensitive and effective diagnostic approach, which is very fast and rapid, does not take much time. I don't have to wait for months and days or an hour. So it is, the response time is very fast. And third question that you must ask, that how the nanotechnology is going to help us improve the existing diagnostic approaches. So the answer to this is nano biosensors. So it becomes imperative to understand what are nano biosensors if we want to understand that how nano biosensors are going to help. So we will talk about what are nano biosensors and how do they work. Nano biosensors are small compact devices. It's like simple, the glucose detector or the diabetes, which is used for people who have diabetes. That's a compact device which measures the biochemical and biological event using different signals. Can be electronic signal, can be an optical signal, can be a magnetic signal, and they will be using this all through a compact probe. It won't be a very big machine. It will be a very compact system where you will be giving the analyte or the sample, and with that small technology, it will be giving you the answer. So this is one of the advantages using nano biosensors, that they are compact. So if I want to understand better that what are nano biosensors and why they are named so, I can break the term into three parts: nano, bio, and sensors. The sensors are those devices which help to us to monitor the changes that are happening around. So if there's any change, the sensor will give us a signal. So I can say a sensor is a device which will gather the information registered. It will look around कि किसका level कितना है, कम हुआ है, ज़्यादा हुआ है, whether the temperature has changed, whether the compositions have changed, and all the biological, physical, and chemical changes that it absorbs, it will convert into some information, and it will tell, just and say, I have some BP problem. So I have a BP machine, it checks the BP and it immediately tells me that there was an increase in the BP or the BP was shot up. So this is a sensor, a device. The term bio means anything that is related to life. So if I say sensor is a device for sensing the changes, and I say biosensor, it becomes a machine or a device which is going to sense the changes in the human body. And what we are going to sense? We are going to sense those small biomolecules, can be DNA, can be nucleic acids, can be lipids, amino acids, the neurotransmitters that we have in our brain, which affect our moods and also affect several serious diseases like Alzheimer's and Parkinson's. So this is the reason we are calling them as biosensors. Why do we call them as nano biosensors? Nano means any particle or any material which is working in the size domain of 10 raised to the power 9 meters. That means I have sensing elements or sensing entities which will help me to sense the changes in the biomolecules. The size is between 1 to 100 nanometer. So these nanosensors or nanobiosensors become small chemical mechanical sensors which will detect the changes in the environment and will give you information. And this is the reason why we are calling them as nanobiosensors. So why we are analyzing the biomolecules? Because they are acting as biomarkers. Now the question is, why are we using nanomaterials? Why is there a fuss about nano? And people always talk about nanotechnology and nanomaterials. So any material which has a size range of 1 to 100 nanometer, and if you want to visualize, you can visualize that it is 1000th part of any questions during the presentations will be answered towards the end. You can note down your questions and I'll be answering the questions towards the end. So it is 1000th part of your hair. You take your hair strand and you try to break it down up to 1 by 1000th part. That much small is nano. So If I am moving from bigger particle size to a smaller particle size, the properties of the material, just say I'm talking about a sand particle, which is in microns, I break it down into smaller size to the nano range, the property of that sand particle will change drastically, both the physical and chemical properties. And this is the catch where the nano has grabbed so much of attention. There will be enhanced surface area, there'll be quantum confinement, improved electrical magnetic properties, there'll be tunable optical properties. And the most important advantage of these nanomaterials will be that their properties are size and shape dependent. I can simply tune their size to obtain different desired properties. The properties are easily tailored, and this is the reason that why there is a boom of use of nanomaterials in the field of sensing and catalysis, because they are the most suitable candidate available. So on the right hand side, the images shows that they are the electrical properties, optical, magnetic, catalytic, confinement, everything changes as the size changes. Also, I can tune the properties by simply not only changing their size, by changing their shape, by changing their composition, by changing their functionalities, their surface charge, and other parameters. So this is the reason why nanomaterials are being used. So now once we have understood why I am calling it a nano biosensor, what nano, bio, and sensor means, so let's move on to the working principle. So look at the image here. In this case, we have three components. These black shapes are nothing but the analyte or the biomarkers we were talking about. This biosensor has a biological receptor and a transducer. This is the place where I'm going to use my nanomaterials. The nanomaterials will be interacting with the biomolecules and they will be leading to the generation of signal. This signal can be anything. They can be changed in color, they can be changed in chemical composition, they can be changed in mass, they can be changed in the electrical conductivity, they can be oxidation reduction that may happen at the surface of the sensor. Any sort of signal that we'll be discussing in the next slide, any sort of signal that is obtained at the biosensor is transferred through the transducer to a signal processing unit. Now, the signal processing unit amplifies it, processes it, and converts it into an electrical signal and gives us a value. So it's not just sensing the presence and absence, it's also quantifying. So what will happen? This analyte will diffuse from the solution to the surface of the sensor. It will react specifically. It may have happened because of the look at the size, that the triangles may fit, but the pentagons and the circles may not. So it will react specifically, lead to the generation of some change in optical electrical properties, which becomes a signal, and the signal will be then amplified and converted into a readable form. So this is the working principle. So like I said, the main important thing is the interaction of the analyte with the sensing unit. And the sensing unit is something which is made up of different nanomaterials, can be nanoparticles, can be nanotubes, nanospheres, graphene, carbon nanotubes, maybe sheets, and it's a long range. So this interaction leads to generation of signals. Signals can be chemical, optical, electrochemical, calorimetric, colorimetric. So this signal analysis will give us an idea about what has happened and up to what extent. Before we move further, it's also important to understand the characteristic. So whenever you want to have a biosensor or a nano biosensor, the sensitivity, selectivity, this list of characteristic is very important. When I say sensitivity, I want to say that even the slight change was detected by the biosensors. I can say that the biosensor is sensitive to the change in the concentration of the analyte. And this will help us that even the minimal concentrations will be detected by your biosensor. When I say selectivity, by selectivity, we mean to say that the biosensor has a particular choice or has been directed to sense certain molecules. Because whenever you give your blood sample or urine sample, there are so many analytes which are present, but I don't want to check anything but urea. So how my molecule is selective to urea and will not be interacting with the other molecule is called selectivity. The third thing is stability. Whenever I want to use the sensor for a continuous time, so whether it is stable or not. The detection limit is the minimum amount of the concentration that can be detected by the sensor is called the LOD, or limit of detection, which is very important because, like I said before, in the early phases of the disease, the concentration of the analyte is very low. And I want to monitor that even the minimal concentration. So I want to have a LOD in the lower range. When I, Hanji? It's requested for to the participants, please mute yourself. Then the next thing is reproducibility. Once I did the testing, it was showing 70 ppm. Again, I did the testing for the same patient, it was showing 80 ppm. Again, I did, it showed 65 ppm. That means the results are not accurate. So I want to develop a sensor which has a reproducibility, or the results should be consistent even after repeated measurements. The most important factor after that is response time. How much time does it take? Does it take hours? Does it take months? Because there are certain testing or diagnostic techniques which take days. We will develop the culture, it will take some 4 days for the test reports to come. So what is the response time? In the case of Nano Biosensors, the time is from few seconds to few minutes. The analysis is done immediately because we have a compact system and the signal has been amplified and transformed into a readable form. So it becomes very quick. Range and linearity is depending on the concentration of the analyte, which is again an important parameter. So whenever you work on biosensors, it becomes important to check all these parameters. The classification of biosensor has been done based on the transducing element and the biorecognition element. Like I said, now from the complete biosensor, I will be just looking at two things. One, what is the analyte? Two, who is analyzing that analyte? The transducer part or the nanomaterial part? So if I look at the biorecognition element, it says what I am actually recognizing, whether I'm looking for an antibody, a phage, DNA, enzyme, or a biomolecule. So what sort of analysis I'm doing, what sort of analyte has been given to me. Based on that, I can say it's an antibody biosensor, or it's a DNA biosensor. It's an enzyme nanobiosensor. Based on the transducing element, I'll check that what response the transducer has given to me. If there was an oxidation reduction happening between the interaction of the analyte with the transducer, I'll say that I have an electrochemical signal coming out of it, or it's an optical, or there was a change in mass. So based on the different types of principle of detection, we have broadly classified them as optical sensors if there is change in light intensity. It can be an electrochemical sensor if there is a change in the electrical response. It can be a piezoelectric sensor if the mass is changing, and if there is change in the temperature at transducer, I'll call it as a calorimetric nano sensor. So the name will tell that what sort of signal it is going to measure. So there is a complete list, and the biosensors have been named based on what they are doing. So if you're going for a DNA sensing, DNA biosensor. If you're going for a wearable biosensor like you have in your hands, these days you have Apple iWatch, which will monitor your ECG, which will monitor your pulse rate. So it's a wearable. So people wear those biosensors, we call them as wearable biosensors. So some important categories of the sensor which I have mentioned here is one, electrochemical sensors, which is going to detect the signal at the interface of the electrode, whether there is transfer of electrons happening or whether the transfer of electron has been hampered. So that is what we discussed in the electrochemical nanosensors. When I am using any carbon-based nanoparticles, just say graphene or carbon dots or carbon nanofibers, I will term it as carbon-based electrochemical sensor. There is a reason why people prefer using carbon-based nanosensors is because they're highly sensitive. Their electronic properties are very suitable for not only the detection of biomolecules, but gaseous compound as well. I mentioned in one of the slides that there are certain small chemical molecules which can act as a biomarker, which are gaseous in form. So if we go for using carbon-based electrochemical sensors, there's a wide range of analytes that can be analyzed. Similarly, I have nanowire nanosensors. Each category has its own importance. Using the nanowires, will have ultra sensitivity and ultra selectivity and a direct detection of those chemical and biological species, which may not be possible in the case of sheet, because I said not only the size but the shape of that material is going to affect the property. So look, for the same material, if I have a cube, a sheet and a wire, the response will be different. The response for the nanowire will be the best. We have piezoelectric sensors, which are measuring the mass to electrical changes. We have DNA-based sensors, which will be detecting the nucleic acids and the direct proteins. We have surface enhanced Raman scattering sensors, which can even detect the molecule to a single molecule level. Even if in the complete solution given to you, there is a single molecule of the biomarker that can be used with the help of Raman scattering nanosensors. We have aptamers and enzyme-based sensors, which I will be giving examples in the case study for a better understanding. Now we have discussed that what are nanobiosensors, what are the principles, and what are their classifications. So why nanobiosensors are getting more attention and are of much interest is one, because of their unique properties, magnetic properties, optical properties, electrical properties, because they are in the nano range. They are showing confinement into a smaller size. And this confinement leads to extra surface area, high surface to volume ratio, higher catalytic properties, higher surface active sites. And that reason enables them to not only sense the biomolecules, I'll say these nanosensors have been explored for complete range of analyte, where nanobiosensor is one important because it is related to the human health. The other advantages of using nanobiosensor is it's inexpensive compared to the conventional techniques where there were big machines, there were complex procedures and steps, like for going for a hemoglobin test. They take your blood, they add it to some solution, then they do some 2, 3, 4 steps, and then they tell your hemoglobin, what is the hemoglobin or the complete blood count in your blood sample. But at the same time, if you want to know how much blood sugar is there, there's a small strip and a machine. So when I'm using a nanobiosensor, the analysis has been simplified. The cost has been cut down. So these are cost effective. These are compact. And not only the glucose sensing, they can even detect bacterial infections, diabetes, and the lung cancers, because every cancer has certain biomarkers. And if I can design a sensor or a nanobiosensor specific to that biomarker, I can sense it at the early stages. These nanomaterials will have tremendous impact on the sensing applications because of the improved selectivity, sensitivity, and the response time to the biochemical analytes. So these are the characteristics which I mentioned in the previous slide, that it should be selective, it should be sensitive to the slightest fluctuations, it should have a rapid response time. This is the reason why nanobiosensors are being used and are of great interest to everyone. Now, to understand better, let's go to some examples of nanobiosensors for disease diagnosis. The first case is neurodegenerative disorders. So when I talk about neurodegenerative disorders, it's like the efficiency and the quality of the function of the brain is decreasing because of certain changes happening inside the brain. So because of the rapid advancement, urbanization, and the race towards Industry 4.0, we are on the verge of breakdown. And since we had experienced the pandemic, there are many cases of depressions, anxieties, stress, memory loss, and many. Also, there is a change in lifestyle, which has taken a toll on the health of majority of the people population. We have that 9 to 5 culture. We are so busy with the work that we don't find time to regenerate our brain. So this has also led to a neural breakdown. So for this reason, the neurological disorders have become most prevalent disorders, and it has ranked the second most serious disorder after the cardiac arrest. So according to the statistic, there have been a 70% hike. And why there is these disorders are happening? These disorders are happening because of the fluctuations in certain chemicals in the brain. And you must have been taught in your junior classes that there are certain molecules which are called neurotransmitters, and I have listed certain neurotransmitters along with the normal range. If they're present in this particular range, it is fine. If there is any change in the concentration, there will be a change in your behavior, in your health, and this will be called as a neurodegenerative. And to name a few, depression, mood swings are the slightest changes. Parkinson's, dementia, Alzheimer's, schizophrenia, these are all the diseases which have been caused by the fluctuations of these neurotransmitters. So this slide shows that at the synaptic left, we have different synapses. At the neurons, these green molecules are the neurotransmitters. So when these neurotransmitters are released into the cleft, there are receptors on the second part. They go and attach to the receptors, and then the signal is generated. Just imagine that these green neurotransmitters decrease in the concentration. So this attachment and the interaction will decrease, and the generation of the response will decrease. My functions, my thinking abilities, my motor abilities will get affected. So this is what happens when there is a fluctuation or a change in the neurotransmitters present. So like in the previous slide, we mentioned the neurotransmitter, which is dopamine, which is of great interest because it is electrochemically active. So when two electrons are taken away from the dopamine, it gets converted into dopamine quinone. So this is an electrochemical reaction where oxidation reduction is happening. So this becomes the basis for the detection of dopamine. And this is one of my paper where we have worked on the dopamine sensing. So we prepared an electrode where lead sulfide was coated, and this lead sulfide acts as a catalyst for the conversion of dopamine to dopaquinone. So just say there are 5 molecules of dopamine present. So those 5 molecules will get converted into 5 molecules of dopaquinone, dopamine quinone, and there will be some consumption of the electrons, and I will monitor that consumption or the generation of the electrons. So we look at the electron transfer that is happening, which gives us an idea not only about the presence, but about the quantification. Like this black one is showing when there was no nanoparticles. When I coated the nanoparticles in presence of dopamine, it gave a significant current response. That current response was because of the electron transfer. So this is how dopamine can be sensed. Then there is one more example where people have used titanium carbide or MagZine for the sensing of, again, dopamine. So they made an electrode, they coated the electrode with the sensing material, and the sensing material leads to the interaction and cause the oxidation. And when there is electron transfer happening, that electron transfer is then recorded. So that transfer of an electron becomes a signal. So a chemical reaction is happening, an electron is getting transferred. So I will term it as an electrochemical biosensor. In this case, they performed differential pulse voltammetry, and look, the blue line shows when there was no dopamine present, and in the presence of dopamine, it was giving a big peak or a big signal indicating that some dopamine is present. It will help us further to quantify how much amount of dopamine is present by further detailed studies which are done in the papers. Now, dopamine was an electrochemically active molecule, but there are other molecules which are not electrochemically active. Then how do I detect them? आप बोल रहे हैं डोपामीन को तो आप इलेक्ट्रोकेमिकल रिएक्शन करवा के डिटेक्ट कर सकते हैं, लेकिन व्हाट अबाउट गाबा, व्हाट अबाउट नॉन अपिनेफ्रिन, व्हाट अबाउट एसिटाइलकोलाइन, वो तो इलेक्ट्रोएक्टिव नहीं है. So there are certain alternate strategies that have been taken. Look, the first case that I showed you was direct detection. The second case is aptometric and enzymes enzymatic sensor. So what I will do is I will attach a specific segment which will recognize your non-electroactive molecule and will be responsible for the change. So now the change is not because of the molecule, but because of the interaction of that molecule with that specific segment. So I have a linker here, which will help me to identify. Similarly, I can use an enzyme. So that enzyme will interact with it and will create certain signals. So we have termed them as aptometric sensors and enzymatic sensors. And because of the understanding of the chemistry and biology, what people have started doing is, instead of enzymes, they have started using mimic molecules or chemical molecules which act as enzymes and will help you to detect the non-electroactive neurotransmitters. This is one example where they have used a gold electrode. They have coated it with graphene oxide, and that graphene oxide has been further coated with orthothalaaldehyde and an alkyl thiol. So when this OPA and Me is attached to this graphene oxide, the GABA has a preference towards OPA. So GABA may not interact directly with the graphene oxide, but this aminobutyric acid or gamma aminobutyric acid will interact with the OPA molecule and will give a signal. So this is what they're saying. If I was not modifying or not adding the OPA, I was not getting a characteristic signal. It was 0. The moment I have added OPA and Me, the interaction changes. So what we are going to do is, in Nano Biosensors, we understand the interaction of these analyte with the molecules, and we modify and tailor them, and we try to get a response out of that because of their interaction. So this enzymeless electrochemical biosensor, where they're using OPA in place of enzyme, they have used it for GABA detection, which is very important. And they have shown that the durability and the efficacy was 96.4%. This is one more advanced technique where the researchers are moving on, in which what they are saying is, just in case someone has a neurological disorder and we are administering him with some medicine. हमने उसको 5 दिन दवाई दी और फिर हमने उसको बोला, okay, then come back to us, we'll test again and then we'll change the dosage. In this case, what will happen? There is a closed loop system. They have attached some in vivo monitoring, which will at that site will monitor what is the level of the neurotransmitter. Just in case if the level shoots up, the medical administration जो दवाई है वो automatically ज़्यादा release हो जाएगी. The moment there is a decrease in the level, the signal will be sent to the medical system, and then the dosage will be decreased immediately. So this is the process that has been done. Till date, I was doing the analysis outside the body. Now we are doing the analysis inside the body, and we are trying to generate that all the minimal fluctuations can be noted at the site itself, such that I can control the release of drugs that is being administered to the patient at the same time itself. So this is one of the advanced techniques which are being explored by the nano biosensor scientists and the R&D people. Now the most common example you people know is glucose detection. How does nano biosensors detect glucose? Is by a simple reaction. They say that if you have a glucose will react with oxygen in presence of certain enzymes and will get converted into some form of gluconic acid. Along with that, some H2O2 will be generated. If I can sense this generation of H2O2, I'll say, oh, there is glucose present in the blood sample. So this H2O2, again, it's an electroactive, it's an oxidizing agent. So it undergoes reduction and it shows a response. So this is how there was a substrate, got converted into product like glucose to gluconic acid. Along with that, there was generation of H2O2, and that H2O2 gave a response. And this is how we are going to go for glucose detection. And this is the principle that is being used. This is an assembly of the first generation glucose sensing unit, which was developed by Clark in uh, in early 90s or something. So there was a glucose, there was a catalase enzyme, which is glucose oxidase. Then there was a cellulose acetate membrane. So all those changes which are happening, we're just saying, can I detect those changes? Can I sense those changes? I will be able to sense the level of the glucose present in the blood. This is another example where people have used a nanomaterial, a copper-based nanocomposite system. They have made an alloy. To alloy to they have made a nanoporous systems, and then they have made a composite bidirectional growth. This composite was when coated to this testing strip. It is very small. I hope most of you have seen those glucose testing strips, which are very small. So this strip becomes that area where that change will happen from glucose to gluconic acid, and where H2O2 will be generated, and that H2O2 will be further reacting and giving an electrochemical response, and we will be able to detect the glucose with excellent repeatability and stability. So even small low detection up to 200 nanomolar has been detected by this nanosensor. So this is the domain where you can work by changing the nanomaterials. 2, as a biologist, you can understand the interaction of the biomolecule with the nanomaterial. 3, as a physicist or as an engineer, you can work on the electronics, the conversion of the signal to the measurable detecting unit. So there is involvement of all the domains of science. This is a repurposed glucose meter where they have used different approach. Because of the short of time, I won't discuss this, but this is one important thing. I hope you remember that I mentioned that there is some amount of gases which are present when we exhale, and that helps us in detection. So this is one technique that has been recently uh, uh, found or invented at the State University at New York, where by just the electronic olfaction, or by just the air that we are exhaling, we will be able to identify if the person is diabetic or not. There's no point to prick your finger and give a blood sample. Even with the air you are inhaling and exhaling, we will be able to do that. And how is it possible? All these developments are possible because there are people who are involved in the development, who are understanding the process, understanding the interactions, and making it better day by day. Another important example is quinolone, which is an indirect way of preventing disease, because there are many people who are non-vegetarian, they feed on meat, and this meat is coming from the animals who have been administered by certain antibiotics. So those remnants of the antibiotics in the meat may be transferred to the human body and may be responsible for certain disease, and these are called quinolones. So even these quinolones which are being injected to the animals, or the feeding cattle and other things, these can be detected with the help of nanomaterials, both optically and electrochemically. So they will have a higher sensitivity and a rapid response, and we will be able to detect them. The most important case is cancer, because initially now we have techniques to detect the cancer at early stage, but initially what happened, the cancer was always detected at a later stage, and the only alternative or the option they had was chemotherapy, which was not successful at that point. So in this case, when there is cancer or cancerous cells in the body, so there is a embryonic antigen which is released into the body. So if I can detect the CEA molecule in the patient, I will be able to analyze and provide a report that there is onset or starting or onset of the cancer. So what they have done in this case is they have taken a magnetic bead or a magnetic nanoparticle, to which they have attached certain antibodies. These antibodies are very specific to carcinoembryogenic antigen. So there will be an antibody antigen interaction that will happen. And then there is an antigen CEA. These nanomagnetic beads were coated on the graphene. So when this was done, the loss of the conduction which happens generally in the case of graphene was prevented. So the moment there is any antigen or any HRP protein which is present will get interacted, and when it interacts with the magnetic bead, there is a change in the current response, and that current response will indicate towards the presence of CEA. That means the onset of the cancer, depending on what concentration of CEA is present in the patient. Another case, so it is also said that there are certain biomarkers called folate receptors, which in presence of the cancerous cells, they overexpress themselves. So if someone is having cancer, there will be an extra concentration of the folate receptors. And someone who is not having will have a limited concentration. So they use indium titanium oxide. And when they're using indium titanium oxide as the sensor, what they observed, then in the absence of folate molecule, there was a transfer of electron happening. But the moment the folate receptors were coming, they were binding to the sensing unit, and the transfer of electron was hampered. So this is one case, there was a signal reduction. So this reduction in the signal or disappearance of the peaks was a signal, again, was an indication that there are folate receptors present. And the extent of decrease, again, it is a linear decrease. So the extent of decrease will tell me how much amount of folate receptors are present. So with the help of this, even they can detect the cancer at a very early stage. There are other applications of biosensors as well. These sensors can be used for soil quality monitoring, food quality, pathogen, environmental monitoring, the detection of the pollutants and the toxic gases. We are discussing about disease detection. So a lot many applications are out there for these biosensors or nano biosensors. Now, the most important thing, if someone hears about, okay, there's something called nano biosensor, how I can be a part of this work, or what are the career prospects and opportunities which are lying out there if I want to pursue the career in as a nano biosensor scientist. Because nano biosensing is an interdisciplinary field. It involves biology if I want to understand the biomarkers. It involves chemistry if I'm talking about the interaction of the biomolecules with the nanomaterials, which are ultimately chemicals. Even the biomarkers are at times the chemicals. It involves physics when there is generation of response, electrochemical signals, generation of piezoelectric response. There is engineering and nanotechnology, which is involved at the same time. So it is an interdisciplinary field. So it holds ample opportunity for the candidates, even with the diverse background. I am from bio, I can find a job. I am from chemistry, there's a job available. If you're from physics, there's a job available for you as well. So from junior executive to the scientists, research engineers, and R&D scientists, there is a long range of opportunities which are available in this field. And owing to the recent pandemic, which we have experienced, the corona and now the monkeypox, there is a huge demand of people, of the people who are working in the sensing domain, because it is very important to detect it on time so that it can be treated immediately and the contamination can be prevented. The opportunities, now anyone who has the knowledge of biosensors, be it a student of chemistry, bioelectronics, engineering, you will have jobs in academics. If you want to go to teach in some higher education institute, maybe you want to be a professor, there are opportunities. You can go to research and development. There are different biosensor companies, which I'm going to show you. There's industry where they're working towards the development of these devices. Again, device design, microelectronics and engineering, there are ample opportunities available in this field. What is the eligibility? You can be a diploma student, you can be a graduate, postgraduate, engineer, or a doctorate. You will find a job in nanobiosensor. Any stream, be it a biology, life science, chemistry, physics, biotech, nanotech, because we're using nanomaterials, or BTech in EC, and CSE, will have opportunities. What is required? It is required that all the participants, all the candidates should have a basic knowledge how the nanobiosensors work. What is the part of the biology? What is the part of the chemistry? How the chemistry and biology are working together and giving the signal is very important. So these are certain excerpts which I recently found on internet about the job opportunities which are available. not only in India, but worldwide. There's a Biosensors International. We have dynamic biosensors, which have, again, lot many opportunities available around there. These are the recent, you can see that it's July 2022, 12 July 2022. These are the research and development technician, the senior chemist for process development, senior biosensor scientist, principal scientist in Texas, in Singapore, and there are n number of jobs which are available. We have universal biosensors, we have SD biosensors in Manesar, Gurgaon. So there are huge boom in this field. Excuse me. Now, when I was mapping the opportunity across the globe, I was surprised to see that the opportunities are available each and every corner of the just excuse me for a second. So the opportunities are available at each and every nick and corner of the globe, be it India and abroad. Now everyone is interested, okay, there are jobs available. What are the salaries? The salary depends on your qualification, depending on your experience. It may range from 3.7 lakh per year to 25.2 lakh per year. There is a growth in the salary with the time and the experience. So if you're joining as a research scientist at L2, the starting salary can be 8.3 lakh per year. And with experience and upgradation, then it can go to 12 lakh and ultimately even 25 lakh per annum. That is the salary and remuneration in the case of the fields in nano biosensor. Now, if you want to pursue this career, how do I pursue? One, the first thing is always review your personality traits. That what do I want to do in my life? Whether I want to be a scientist or an academician or I want to be an engineer, review that. And based on that, earn the basic education, earn a graduate degree, irrespective of which field, depending on, again, on your interest. You want to go for astronomy, computer science, biology, physics, choose your career path. Accumulate job experience in that domain. And whenever you want to go for such a job for biosensors, because there is no degree in biosensing, you don't have MSc in biosensing, you don't have graduation or BTech in biosensing, you need to obtain certain certifications or some experience in that particular field. And with the qualification, with the experience, and with the app knowledge, you will be able to join your dream job. So this was from my side. I hope you enjoyed the session. Thank you so much for being a patient listener. If there are any questions, I'm open to the questions. There is a link and a there is a QR code. If you want to scan and join the course, you can join that. There are people who are asking for the notes and the presentations and the recordings. I'm not sure. I'll check it with the team from Learn to Upgrade. If it is possible on their end, they will do that. Any questions, you can please type in the chat box. The certificates will be provided by the, again, Learn to Upgrade team on after the completion of this webinar. Any questions related to Nano Biosensor, you can type in the chat box.
Thank you. Thank you, ma'am.
Welcome. Is it possible to monitor our glucose level by using non-invasive glucometer? I just mentioned that recently in New York State University, people are working on that instead of going for the blood sample, you can sense the glucose or different biomarkers, which are biomarkers for diabetes, through the air that you have exhaled from your nose. So we have e-noses coming these days, olfaction or electronic olfaction, we say. So that can be sensed. Yes, it can be sensed. What is the role of biotech? The role of biotech is major because it involves the complete interaction of the biomolecules, and we must understand that how the body is responding, what are the biomarkers which are being generated, what are the concentrations required, what are the concentrations which are off the mark, the interaction of the biomolecules. So biotech is one prime branch which is our desirable candidate in the field of biosensors. What is nanocones? Okay, whenever a material is named nanocones, means because the size or the shape of the nanomaterial is cone shaped. If it is spherical, I'll call it nanosphere. If it is cube, I'll call it nanocube. If it is a wire shape, I'll call it nanowire. So if it is a conical shape, I'll call it as a nanocone. Any side effects of using biosensors? Most of the biosensors are used in vitro, outside the body. We are not administering, except for the MRIs, the imaging analytes that we use are administered. Otherwise, most of the biosensors are administered outside the body. So there is no invasiveness. So there are no side effects of the biosensors. How can I get a dissertation project? Being a UG Tech student, you will have to contact people at your university, and you can approach other people who are working at different universities, and you can conduct internship on this topic. For that, you'll have to find a suitable mentor. What master's time would you recommend for this field? Depending on your bachelor's, if you are bachelor's in biology, I'll suggest go for the master's in biology. If you're bachelor's in chemistry, I'll suggest go for bachelor's in chemistry, because each and every field is very important. A biologist cannot work without a chemist, because you need to understand the interaction. A chemist cannot work without a nanotechnologist, because you need to design those materials. Again, we need engineers for that as well. Can we change the property of a cell using nanomaterial? That depends on the case why we are going to change. Yes, we can do that in certain cases. Use of nanotech, yes. A lot of use of nanotech. Nanotech is that interdisciplinary branch where people from chemistry, physics, maths, and bio go and understand the basics and the nitty-gritties that what is happening at that nano level and how a material will interact and respond. Sir, is the review link going to be Google search page? Sorry, I don't know. The team from Learn to Upgrade will be on. Can biosensors be used in urban farming? I don't understand what you mean by urban farming. But if you're talking about the detection of agrochemicals or certain pollutants in the soil, yes, people are doing detection of certain pathogens and even the agrochemical molecules using the sensors. Biotech graduate, how can I get a job? What do I need to learn to clear an interview? Whenever you are going for any interview, please understand what is the profile, what are the responsibilities. Try to learn those. Whenever there is a job profile, they mention what are the responsibilities that you must be able to perform, the portfolio. So you need to understand if there's a job, just say in Biosensors International, they're saying I want a senior biologist, or I want a biotech graduate who must be able to do this. They must be able to write SOPs, they must be able to conduct independent research. So this is how you can prepare. You can also look into the website and check what they are doing and try to understand what they are doing. That will help you a lot. Biosensors to be receptive for cancer detection, is it the folic acid chemical pathway or chemicals related to different cancer, different pathway? There is a slide in the starting which I showed you, and which is even available on the internet. So there are different biomarkers for different sensors. If there's a lung cancer, there will be a different biomarker. If there's a breast cancer, there will be a different biomarker, because there's a different response and interaction. So different biomarkers will have a different sensor. So I'll make a sensor which is selective to breast cancer. If I want to see whether a person has a breast cancer or not, I'll use that sensor and we'll be looking for that particular biomarker in the body. If there's a biomarker, I'll say, yes, the person has biomarker. So there can be different pathways and different biomarkers for different cancers in the body. The electrons are detected by the unit that we have. We have galvanostat and potentiostat. We have a 3-electrode system, which is complete electrochemistry. We make electrodes, we connect it to a system, we provide a bias, and there's a current response, current to voltage response. That current to voltage response will give you an idea that how much amount of electrons have been generated or consumed, because electron is directly related to the current. Higher the generation of the electron, higher will be the current. Its spelling contains glucose molecules and low concentration, accurate glucose molecules. It is because of the sensitivity levels. We are working on the sensors which can sense to a very minimal concentration changes as well. That is the idea of using biosensors. Nano biosensors fast diagnostic about other than laboratory correct or not. Again, there is a big debate between the conventional diagnostic approaches and the modern diagnostic approaches. What it has been observed that, again, like I said, if you want to check whether this biosensor is a good one and I should go for it, check the selectivity, sensitivity, response time, and reproducibility. If it is matching all the criteria and the characteristic, it is obviously better than the laboratory reports, and it will take time for these biosensors to reach the lab. Role of microbiology. Okay, in microbiology you have virus and bacteria, and these virus and bacteria are pathogens which lead to generation of antibodies and antigens. So again, that will help you if you are from microbiology. Biomedical engineering, want to go for sensors and clear about your experience knowledge. Suresh, you can contact me. You know my name, you just search my name on Google, you will be able to contact me through that, and you can contact through Learn to Upgrade team also. If you have any query, you can contact through them to me. What are the recent areas where are being developed? Cancer, insulin detection. So about biosensors, the recent areas, the recent example was corona. The test kit that was being used, which was giving a direct result, was a recent example. Okay, this is the end of session. If there are any questions, you can type and leave it. The team from Learn to Upgrade will pass on those questions to me, and we'll find a way to answer those questions. Thank you so much.
Thank you.
Welcome.
Thank you, ma'am.
Thank you so much, ma'am.
Welcome.
Thank you, ma'am.
Thank you,